Now this picture here, I don't know if you know this area of town. If you, these cars are traveling down Richmond Street, and it's at the corner of Windermere. I used to walk this way home every day. So, and today we'll talk about this area, and it's a very prominent area, focusing in on motion. So remember, I mentioned that in the back of the brain here, there are a couple of dozen, 3 dozen, perhaps maybe 4 dozen, that keep multiplying, areas that are sensitive to motion. And when you, they're like um, a, a, a theater filled with screens. And each one is sensitive to some unique aspect of what you're seeing. Not 36 areas involved in motion, involved in vision. Some of them are involved in motion, and this is one of the more prominent ones. Now, this area is called Area MT, and it's called Area MT for a strange reason. It was first found in the owl monkey, and there it was located in the middle temporal gyrus. In humans, it's located here. You see this thing. This whole thing is the temporal lobe. This sort of, you can imagine this is a thing sticking out of the side of the brain. That's your whole temporal lobe, and under here is where all your vision of objects is, and particularly faces. So this is a fold in the bottom of that temporal lobe, the inferior temporal sulcus. And as you head up here, at the very end of it, you find this area MT+. But of course, it's not called IT+, but because we started naming it as MT. And what does it do? Well, when you have MT, you see this. Everything is moving smoothly, and we can see objects. We can see there's cars. Now, if you notice here, there's a car stop. If you watch carefully, there's a car stopped there, okay? Now, without MT, you can't tell that there's a car stopped there because everything seems to be jittery, being stopped. So this is what your brain would see if you didn't have area MT. Everything would be a bunch of stills. When you poured water into a cup, you would just see it's one still, and then another still, and then it's overflowed. It'd be very hard for you to tell that the water is rising in the cup. You tend to overfill your coffee cups with hot water. Now, if you didn't have anything else but area MT, you'd see this. So this is what MT sees. Surprisingly, it doesn't see very well. It sees in black and white because, again, it's getting most of its information from the peripheral retina, which is mostly your rods. And it's also, the information is coming after lots of conversions. So the receptive fields are very large, and you don't see in detail. But you can tell that something's moving. You can tell that there are all kinds of objects moving around. If I had blurred it enough, you wouldn't be able to tell them that they were cars. So it's kind of remarkable that in one area, this is what you'd see if you were sitting in that area and watching it. And if you were in another area, somewhere in your what stream, you would see this, because the what stream has no sense of motion on its own. But somehow your brain puts together what this screen sees and this screen sees, and your brain as an entity sees these objects moving. So somehow it tags this motion of this car with this still of this car, and it does this on the fly at an amazing speed. So without noticing, you perceive these cars as moving, without noticing and without effort. Now, we saw that that information comes into your layer 4C, and that it heads up, for example, and goes to these blobs. And that's where you have your double opponent cells. We saw also that it goes off to these edges, and you've got your interblob area, all these sort of spokes of a wheel, and you sense what orientation a particular little segment of a line is. And both of these have very high visual acuity. In contrast, the area that feeds MT comes along this pathway. It goes first to layer 4C, but then to layer 4B. And again, many of these cells are binocular, so they receive information from both eyes. And so information here can see in depth. You can see how far an object is from you. And from here, the cells feed either MT directly, so a cell from here will go straight to MT, or it would go to V2, then V3, and then MT. Now, this is an example of a cell that's sensitive to motion. Okay? And what you can see here is, as soon as this cell fires, this cell then feeds, let's say, area MT, and you have this sense of motion. So it's all dependent on this cell firing. Now, to get this cell to fire, all these things have to arrive at the same time. And in order to arrive at the same time, this axon has to be a little bit longer, because this was the light hit this receptor first, and this a little less longer, and this is the last one to be hit, so its axon is the shortest. If it's arranged this way, all these action potentials will hit this at the same time, and the cell will fire. We'll watch that again. Now, in contrast, you can see when light goes in the opposite direction, it activates each of those neurons in sequence. But because these axons are not of the right length, the action potentials reach this neuron at different times, and the cell does not fire. So you don't have any sense of motion from this array of cells in your area MT. Now, in some species, like birds, this type of circuit is in the eye. In other species, such as us, something like this is in your V1. Okay. Now, what does the brain do? V1 gets these simple and complex cells, and some of them sense motion. But they've got a problem. Now you look at these lines, and they seem to be moving in different directions. Okay? But let's take a look at but this is what your V1 cell would see. The simple cells see things through a receptive field, and the receptive field is limited like this. So if you see the whole thing, you can see that this line is not moving this way, but actually moving this way. This line isn't actually moving that way. This line is actually moving that way. And this line is not moving up, but it's moving again. So all these three lines are actually moving in the same direction. And yet when we see them only through the aperture, we see them moving in different directions. So the brain is doing something on these lines, assuming something about these lines. And what the brain is doing is assuming that the line is traveling perpendicular to the line. So if it doesn't see the ends of the line, it assumes that this is the way it's going. Okay. So it's making a best guess. And one way that it accomplishes this is because remember these simple cells feed the complex cells, and these simple cells are all sensitive to a line that's in this direction. And so if all three of them fed this complex cell, of course these lines would have to move in this direction to activate this complex cell. So the circuit is built to be triggered by lines that are perpendicular to the aperture. Now, sometimes we don't do that, okay? If it was that we just looked at the aperture, this line would not seem like it's going down now and horizontally now. It switches from down to horizontal when it hits the different parts of the L. In reality, it's moving sort of in this oblique angle. So there's another factor that the brain tries to use to decide which way something is going. And that other factor is that it treats this line sort of as an object. And it assumes this line ends someplace. So it makes assumptions about where the line ends. So here it can see the line. Here it can't, but it assumes that the ends of the line are always there. And in this case, it assumes the ends of the line are moving. Can you see those tiny little circles at the ends of that line there in the L? I haven't drawn them big enough, I think, to accentuate them. Okay. Now, I mentioned that I think your perception of motion lies within area MT and not in V1. But how do I go about proving that? Well, there was a scientist way back that did an experiment like this. So he measured from a cell in V1 and later we'll see in area MT with different patterns moving in front of the eyes. And recall that a good stimulus to use is these lines when you're trying to activate cells. Now, if you used a set of lines that went up to the right, that set of lines didn't seem to do much to the cell. If instead I used this set of lines, a horizontal set of lines, again, that doesn't do much to the cell. So this cell prefers this line, lines that are moving down and to the right. Now, if we move the electrode over to area MT, we, by luck, managed to find this cell doesn't like lines up to the right, doesn't like lines moving to the right, but does like the same direction as we found the area V1 cells. So you can see that this stimulus would not be able, using that stimulus, we couldn't tell whether, in both cases you perceive motion, but it's the same motion activated both V1 and MT. So it's not a good thing to use if you want to tell which it is. One good thing to use is this stimulus here, a plaid stimulus, or plaid stimulus, I'm not sure exactly how to pronounce it. But you can see here, first we have lines moving up, green lines moving down, blue lines up, but the combination of the two moves horizontally to the right. Okay, so when it's moving horizontally to the right, like this, there is physically no line moving in this direction. It's an illusion. There are only lines moving up to the right, another set of lines moving down to the right. Okay. So and yet you perceive this set of lines moving horizontally. So what if we use that as a stimulus? Okay. So you see here a cell in V1 firing for the stimulus. Okay. Now remember that cell in V1 was sensitive to a line moving down to the right. Why is it firing? Well, because there is one of the set of lines is moving down to the right. Okay. This direction is these lines moving this part of the pattern, lines that were drawn this way. The ones with what would be displayed as the green set of lines before. If now we go to this pattern here, I hope. Yeah. Okay. Again, if we go to this pattern here, yeah, okay. So that was moving. Okay, so we take the flat pattern. We can make move down, we can move it in this direction, or this direction. Okay, so now the flat pattern appears to be moving to the right. And again, the empty cell is not firing, but the V1 cell is firing, because there is still a set of lines moving in that direction. If we move this way, in contrast, we now light up the empty cell, but the V1 cell is silent. Okay? The empty cell is firing because, yes, you perceive the stimulus moving down, even though physically there is no line moving in that direction. The V1 cell is silent because the lines are moving either to the right or down. There is physically no line moving down to the right. Okay? And finally, if we use this stimulus, again we light the V1 cell because now there is physically something moving in that direction. But when you look at it, you just sense the object is moving down, not down to the right. So MT fires when you sense something moving in its preferred direction. So this proves that your sense of motion is whatever MT tells you it is, and not necessarily what V1 says it is. Because V1 would not sense this directional motion. The other thing about MT cells is that they're sensitive to attention. So again, we're recording an MT cell, and you can see it's not very sensitive to things moving down, but it is sensitive to things moving up. Now let's complicate the matter. Let's have it have two dots moving up and down. But we pay attention only to one of them. We pay attention to either the green one or the blue one. Now, you can see that the cell fires now when something is moving up, and we're paying attention to the blue dot. Now it's firing again, but we're paying attention to the green dot. So it's sensitive to whatever we're paying attention to, and it's firing and it's moving in the correct direction. Okay? So it doesn't matter when we're paying attention to the other dot, it doesn't matter what the other dot in the scene is doing. Okay? If we're paying attention to this dot, it doesn't matter what the green dot is doing. If we're paying attention to the green dot, it doesn't matter what the blue dot is doing. Okay. Now, area MT is, again, has a retinotopy to it. That is, the eye is mapped out at different portions of the eye. Again, it'll be largely a peripheral eye, because that's where it gets most of its information from. Now, so it's made of columns. Now, if you take one of those columns and look at it in more detail, you see this pattern. So within one column, that is information from one part of the retina, that information is being analyzed in all this multitude of ways. Now the arrow direction says, okay, some neurons over here are sensitive to motion in this direction. Some other neurons over here are sensitive to motion in slightly a different direction. The colors indicate what distance the object is, the moving object is. The ones in green say it's something farther away. The ones in red are something that's nearer to you. So we can see that all these different directions and these different locations in depth are being mapped here. In addition, within any one of these little mini columns, you have many, many cells, and each one is activated for a particular speed. Remember how those series of dots at the activation of that neuron early in this lecture looked like. So again, if you see something moving, it's moving in a particular direction, a particular depth location, and at a particular speed. All that is determined by the firing of one little group of neurons. Now, MT plus is in turn divided into area MT and MST. Okay? And this area MST has two parts to it, an L for lateral and a D for dorsal. Now, the L, MSTL, the lateral part, it likes objects that are moving in your visual field. Okay? So if you're following a tennis ball, if you're looking at this plane that's moving, a bee that's flying, a bird that's flying, something small in something else that's moving beside you. Okay. Right now, if you're looking at me, it'd be this hand that's moving. If you're looking at your fellow students, they'd be shifting their heads or looking in different directions. In contrast, this area MSTL, TD, is sensitive to you moving. Okay? So L is for objects moving, anything but you. And D is for you, what you would see if you were moving. And so what they are sensing, so if you're riding down the road here, you would sort of sense that you're moving. You'd be looking here in the middle, and your entire visual field would be filled with things moving. So both things in your left visual field and your right visual field. And this pattern that you see will tell you that you're moving in this particular direction. And if the pattern changed, you'd sense that you're moving in another direction. So the cells in this area are in some ways unique because they're getting information both from one side of the eye and from the other side of the eye. Of every area that we studied up to now is just getting things from the opposite side of the eye, okay? From the contralateral visual field. The only other cell that we found that does this was your cells in your the cells that recognize faces in the fusiform face area. So to recognize a face, you have to look at both sides of the face, and they also get information from both LOCs. In this case, this MSTD is getting information from this MT, but also from the one on the opposite side. Okay, so often when you're stopped at a red light and there's a car in front of you, and the car in front of you starts to move, you sometimes sense that you're moving backwards. Or if you're in a, say, subway car, and a subway car beside you starts to move, you sense that your subway car has started to move incorrectly. So because the information in these cells is filling the complete visual field, you often sense that you're moving when you're not. Now, different patterns will cause you to sense that you're moving in different directions. So, for example, here you sense that you're moving backwards. This pattern will tell you that you're moving forward. This pattern that you're going to the right. This pattern to the right. This pattern to the left. And you can also get this pattern that you're flipping upside down. You can imagine that there's almost an infinite amount of patterns that you can have, that you get combinations of these. And each of them would tell you some unique direction that you're going into. But to get these patterns, again, you need to look at what's happening in your entire visual field. So both information from the left and the right are required in these cells to complete the pattern. So in MSTD, you have cells then. One particular cell will code one particular direction. Another different cell will tell you that you're moving in another direction, and so forth. Now, this is another type of motion that you see. This is called motion parallax. Now, you could compare this and this. This seems 3D-like, I hope, to many of you. But it's actually, it's a flat screen, and you're seeing it with the same visual scene with both eyes. There's no difference between the two eyes. So why is it that you sense that this object is three-dimensional? This is the backyard of my garden. or not my garden, my wife's garden. She's the gardener in the family. And this is would be what the flowers looked like a month ago. Well, we looked at before your ability to see in 3D, and that was the stereo vision, which worked on the disparity, the difference between what one eye sees and the other eye sees. And then there are cells in area V1 that compare the two and light up per particular disparity. Well, if you get to about more than an arm length away, this disparity becomes so tiny that there's almost no difference between the two eyes. So to measure differences where things are for larger differences, you need something else. And that something else is this motion parallax. Now, you can see here the eye is pointing straight forward. And if you compare what this square and this red circle are doing at the back of the eye, you can see that the one that's farther is moving slower across the back of the eye. Because as the head moves back and forth, it makes a smaller sweep across the eye. So what the brain is doing is comparing how things are moving relative to each other. And from that, it can measure distance. I don't know if any of you spent a lot of time watching chipmunks before they leap. They tend to bob their heads up and down. And that gives them this type of motion parallax. And so they can tell how far away where they hope to land is. So they're using motion parallax to judge distances. So anyways, this gives you a depth attribute to that motion. Now, one of the complicating factors is that the eyes move. So often, when you're looking at something like this, and move your head back and forth, you'll be staring at one of these objects. And when you stare at one of these objects, your eyes pursue it back and forth. Your eyes keep pointing to it. So here you are, you're looking at something. This is the scene. This is what your eye sees. And again, this is what your eye sees. This is what your eye sees when you're looking at the purple flower. So these are objects close to you. Okay? When you're looking at the purple flower, you can see that the purple flowers are moving less than the yellow flowers. If, in contrast, you're looking at the yellow flowers, you can see that they move less here through this aperture than the purple flowers. Okay? So it depends where you're looking. And here you can see, again, these squares, but now the eye is turning with the motion. Okay? It's always pointing to one of these objects. So in this case, it's pointing at the near thing. And of course, the near thing is then stationary on the eye. It's always pointing at the back of the eye. But the red thing moves back and forth on the back of the eye. In this case, it's looking at the red thing, and the red thing stationary at the back of the eye. But the green thing moves back and forth. So what moves fastest on the back of the eye depends also on where the eye is looking. Now, this being the case, how do you tell in the visual scene what's moving? Okay, if the eye tends to move and it changes what the back of the eye perceives, how do you tell? So here is it the line that's moving, or is it you that your eye is moving? And of course, if it's something like a lion, it's important to be able to distinguish between the two things. So over 100 years, Helmholtz discovered, and it has been proved, that how you distinguish between these two. And you use this thing here called correlative discharge. Your brain puts out a signal for your eyes to move. And a copy of that, so this command gets to the muscles that turn the eye, and your retina goes back and forth. This signal is also sent to the circuit that does this comparison. This circuit also gets information from how much things are slipping on the back of the eye, whatever the back of the eye sees, and sends that signal to the brain. This area here compares these two pieces of information. And this comparison produces a signal which is the object's true motion. So it takes away whatever motion is due to you by doing this comparison, this correlative discharge. So let's suppose this is what happens, that this motion here is because the eye is turning to the right. Well, the image on the back of the eye would cause this image to slip to the left. Okay, so you'd see retinal slip to the left, and that signal would come in here as well. And where signal to the left meets a signal moving to the right, the two cancel each other. And the brain decides, the line is stationary. It's my eye that is moving. And the same thing happens for other kinds of motion. So when for example, when you're walking towards something, there's a correlative discharge being sent to the brain that you're moving. And you can distinguish between this line getting bigger because you're moving versus the line is moving towards you. Very important to distinguish between those two things. Okay, let's just do something fun. Stare at the center of this circle, okay? It'll start turning, and then it'll stop, and see what a face will appear, and see what you sense the face doing. What does the face do? It got bigger, yes. Yeah, exactly. I'll do that again. Okay, let's do it one more time. This time when I stop, okay, look at a face beside you. Whoops, sorry, I did. Hopefully the face beside you didn't move. Otherwise, you compound the illusion. Okay, what's happening here? This is called the motion after effect. And it produces changes in MT. Now, one of the misinterpretations with this is that the thing is moving, and cells in MT fatigue. That's not the case. Cells don't get tired. At least at least if they're given enough oxygen. So one possibility is that the cells adapt. So with time, the brain likes changes, so gradually things slow down and stop firing. And then when it stops, you sometimes sense motion in the other opposite direction, and that's why you see the face coming towards you. The other equally likely possibility is that when you see something moving at a constant direction, your sense of motion gets recalibrated. Okay? You can imagine that you've got lots of cells. This cell senses this motion at this speed, this other cell here motion really fast at the opposite speed. And over here it's 0, and cells here sense motion slightly in this direction, and this is slightly in the opposite direction. So when we have, let's say, a stimulus causing motion to be felt at this speed, you can see that the cells at this speed recalibrate themselves, and more of them sense motion at this speed. This is useful because, let's say you're driving on the highway at a particular direction. You want to have these cells group at the speed that you're moving so that you can sense more clearly small changes in that speed. You're more accurate at determining whether things are moving faster or slower at that speed. Now, what happens as a consequence, watch this yellow cell. This yellow cell fires when something's moving slightly in the opposite direction. When this starts moving, you can see that cell being dragged over to this area. That area is the motion in which cells are still. Okay? Something that's nearly still would now activate this cell. Okay? But it was the cell that was calibrated to the opposite direction of motion. So when it's firing, you sense motion in the opposite direction again. So that's why that face appeared like it was moving towards you. Okay, what's interesting about this? Well, you can see whenever the fragments that contain the image of a lion move, you see the lion. But when they're still, the lion disappears. Okay? So it's motion that's causing perception of the lion. But MT doesn't sense the object, okay? So MT is feeding its information to some area like area LOC. There's MT, and it's feeding information to the watt stream, perhaps through area LOC. And it's telling LOC which fragments belong to the lion based on its motion. Okay? So group these lines together, and you'll perceive a lion. What's interesting about here? Okay? If you look at this, it seems to be a white square moving back and forth. Hopefully all of you are perceiving this white square moving back and forth. But in reality, there is no white square moving back and forth. What I've taken is a square over here that's white, and then shown it over here as a white square. Okay? So here, LOC is giving MT a false signal that something's moving. A square appears over there, a square appears in another place. Two squares, both stationary, give you this sense of motion. Okay, so we see that MT and LOC are feeding signals to each other. One to tell you which parts of the object should be bound together and made into an object, and the other telling you perhaps something that's an object is moving back and forth. Okay. Last, I'll talk to you about this thing called biological motion. So here you see what? What do you think this is? Is it okay? Is it something alive necessarily? No. It could be a stationary statue being turned round and round, or you're going round the stationary statue. It could, of course, be someone still is turning round and round, but that's from just this you couldn't tell it was alive. If you look at this, you can tell that it's something living. Okay? So what's the difference between these two things? And it's remarkable that the brain can tell this. So in this case, these lines are turning, but the brain can figure out that all these joints, where all these joints are, and although they're changing on your eye, they don't be changing on the object the brain has created. The brain is creating this image of a 3D statue or a 3D person, but it's not moving. It can tell it's not moving. Here it senses that joints are changing in their relative location. Okay, the joints are the hands are moving, the feet are moving. The angles are changing in the scene. These line fragments are changing the relative location, such that it could be a real life object. Now, this type of relative motion can be formed by this figures that's moving, or things like your lips moving. And what's responsible for the sense of this biological motion is this area called STS, the superior temporal sulcus. So we had this thing called in the inferior temporal sulcus, and now we're going over a place in the superior temporal sulcus. And so it's combining information from MT and from LOC and telling you that this is a living object. And this area is useful for things like lip reading. So you can use lips to be able to decode what a person is saying. And you'll see in a moment that you can tell a lot about a moving figure. Okay, we saw that the output from the visual system is divided into two streams. The WARE stream, which comes primarily from the peripheral retina, and it feeds the magnocellular LGN, and then goes to the posterior parietal cortex. We've got the Watt stream that comes primarily from the fovea, goes from the parvocellular LGN, and gives you details of what objects are in the inferior temporal cortex. So in terms of input, the stream we're talking about today seems to be getting most of its information from the WARE stream. Okay, so it seems to be a WARE stream type of thing. It's got poor acuity, it senses motion. But some people call this a WEN stream, a third stream, because it provides information both to the Watt stream and to the WARE stream.